Good afternoon, everyone. First of all, it's always nice to start with a thank you to the organizers of this. It's always nice to have the opportunity to present yourself at these kinds of meetings. As you can see from the title, we have, at least when I made the presentation, I've changed that a little. The original one included capillary electrophoresis, and we still use that in the lab, but that's only for some very specific diagnostic markets that are difficult to run on any other platform. And that's kind of partly leading to the content of what I want to talk to you about. It's my impression that it would be useful to spend a few minutes on introduction, so everyone knows where we're coming from. We would need some background here on the market analysis. It's not going to be as in-depth as the first presentation, but this is mainly about the animal markets that we are working in at the moment. Some of the scopes of the partnership here for today, and then I'll show you a number of examples which are not necessarily, maybe a bit too much detail here, but I just want to make sure we're on the same page here. And then I'll conclude in about 20 minutes. It's different when you have something in your hand to click on. We have been around for quite a long time. There's a number of companies that started in the 80s. We have been growing through the different cycles of the technological developments. What we mainly focus on is DNA. There's one or two exceptions that we still do, but our client base is very wide. Personally, the introduction has been done already, so I'm a geneticist by nature. And the company part has mainly grown by listening to people who have more understanding of that than I do. So for the moment, we have 3 business units in Europe, and we speak to a lot of people here. Our main reason for being here is that the Plant and Animal Genomics meeting has been growing over the years into a global meeting. And it's like coming home here every year in the middle of the winter in Europe. It's perfect to be here in the sunshine for a short while. Our main facility is in the Netherlands at the moment. We have a much smaller lab in Belgium, and we have a medium-sized lab in Germany. And I assume that all folks from North America will be guided with this small section of Europe for your background information. It does mean that we work globally, but still our home territory is Europe. That's at least how we consider it. What we currently are emphasizing is that we do continue to grow into Europe, but with some high throughput internal hardware. We have chosen to do that through a distribution model rather than having local owned smaller representatives by ourselves. And this kind of summarizes it all. We used to stand here as VHL being Dove and Herring Laboratory 5 years ago, and we got so many questions on what that funny name meant that we just decided to go a bit more international. We work across 3 main fields of expertise, but we always point out that we don't work in humans, and that has to do with a lot of the European regulations and all the genetic counseling around that. So we do have specialized ourselves in the rest of the applications. Historically, it's only been animals that we've been working on, and the examples I will show you today are also coming from the animal side of things. But we have grown significantly in the plant side of the business because of the increasing availability of technology. And somewhat of a different type of specialty is the microorganism stuff, which is definitely not the microbiome that we are working on, but this is really dedicated pathogen detection in the various fields that we're active in. And to show you a few examples of how wide that goes, it really extends from the business-to-business side, including governments, up to the business-to-consumer side, where people want to test with their own personal pet on a number of markers. And that will be the main topic of the presentation for today. But the terms that you would find in these fields are the parentage verification and the genetic variants, as they are much better called than diseases and mutations. And there's a lot of growth in there. We do some research ourselves, but we try to work in partnerships and follow literature when it's presented. There's a lot of work done in genetic variants in plants. We have partnered there as well. It's kind of the key how we have developed over the years, and also the reason why we're so in favor of larger partnerships, which makes sense to the community and us as well. The detection of pathogen that I spoke about and the species identification is one of those outliers, but really kind of leading to why we do this. Because if you look back over the last 30 years, there is a continuous need for improvement. And I'll show you some more examples of that later on. But there is, what's intrinsically happening in our field of expertise is that there is a transition in markets. It's no longer a business-to-business market that we look for in the long run, but we are looking for a business-to-consumer market in the long run. It's like ordering your clothes or your shoes on the internet. You would just order your DNA test through a web shop. We send you a sample collection kit. You get the results back uploaded on your cell phone. And that's also where you have much nicer pictures. I'm not sure who has a puppy here or who owns a dog, but it's always nice to show those kind of pictures as well. And much of the developments that are going through in our field are actually coming from the focus on animal welfare as well as the consumption. There was a presentation yesterday here, and if it was correctly, it's either GMO or it's organic. And that's how the world is going to view our field, especially in the field where we are working with the consumers. I don't know if that's the same here, but there's a lot of focus on the animal welfare side in Europe, where there's a lot of government discussion, and it's actually going to the level where currently in Belgium, it's the king himself who's signing a list of markers that has to be tested in every single dog. So it's going to the stage where it becomes really political as well. And that's kind of the background of this particular market. We are, because of all the new developments and all the new know-how in the field, we are currently in a transition where it historically is coming from 1 to maybe up to 100 genetic markers, or it would have been the high end, where you would talk about the 10,000 plus marker range. And we have been looking for a number of years to try and identify a flexible system where you can actually do the intermediate range, where most of our applications are actually moving to at the moment. If I kind of look at how we developed over the past 30 years, we have always tried to partner with small, medium enterprises, because that's kind of the same vision. If you share that for 5 to 10 years, you can quickly develop. With the current technology around, you need to be able to partner with the larger technology providers to the same reasons as the previous presentation was about. So kind of what we look at is the partnering is kind of an answer to some of the development stages of these applications that I just mentioned. And it's a combination of the specific information of the field that we would provide with in-depth know-how, and the continuous development as kind of the technology that I'm going to show you is now in the 4th or 5th generation with a 3rd generation equipment. And that's kind of how we like to work, because then you have a long-term, long-vision co-development situation. So we look at ourselves as being the application partner in this, where we have detailed market information, especially in Europe. That's quite challenging. You have to be around for a long time to actually know the networks, because they are usually amateuristic and not developing too rapidly. At the same time, the routine lab requires much more flexibility in the marker numbers as well as the sample numbers. And of course, to move forward, it has to be cost effective and hopefully cheaper than it was the previous time. So we are absolutely happy that we have been able to partner in this case with the Thermo Fisher side of things for this particular technology. And it's kind of the same warning that the previous presenter also indicated. If you don't have good quality DNA, it's no use to start. And it's especially in the field where we're active, we try to be part of all the working groups around the world that set the standards for this kind of work, because it's, there's a large number of much smaller labs, and they really have a hard time of following up on the developments. And some of them are really still coming from an angle where you can, they think you can do a lot of bad quality DNA into high quality results, which we disagree with. So maybe it's overdone, and it has been partly done already, but I just want to show you the simple basics of the AmpliSeq technology so that everyone is involved. But there's, of course, the booth here where you have much more in-depth information. This technology is based on a system where you can choose the number of reactions that you want to run. And the idea is that in each sequencing well, you end up with one sequencing strand. And because we have currently set this up in our main diagnostic pipelines, it's really important to have high quality. And that's also why I want to show you a few of those examples where if you don't careful, you're not going to make it. The system is based, and this is the AmpliSeq system, is based on a flow system where you push the nucleotides each individually over the different wells. And because every little well actually has its own detector, you will be able to detect the changes in the pH, which is then afterwards leading to the detection of the sequence. It's a 4-step process at the moment. The library preparation takes you 1 day, and then the overnight steps followed by the sequencing the next day will give you the results in 2 to 3 days, depending on how you actually set the workflow. For us, one of the key drivers for this is the scalability. There's 3 different chip types where you can go from smaller number of reads up to the high end, and depending on the number of samples of the project that we want to run this week or today, especially in the business to consumer, the time frame is as important as it was in the previous presentation in a totally different field. It does make sense to look sometimes at the optimal size of the chip. However, for our diagnostics, we always try to do the high coverage because we want to make sure we have the right result to those end users, because they can be pretty nasty on the phone. And currently it's a situation where you can combine up to 384 different samples, as most of you will know, I assume. What we have chosen for the flexibility on this is that on the same run you can combine all the different species of interest and all the different samples that came in this morning or yesterday. We try to get them as much automated as we can, but especially when you work across Europe, you will have a large variety in the sample types provided and a high requirement on all the different steps that you have to do. So currently, I'm only going to show you some results on the feline, but we have 3 products that are already running in the lab, but are in the pipeline to come out. And we call them V1 because there's not been a lot of time spent on how this is going to be named or what the names are going to be. The first version in the canines and felines are really aimed at genetic variants, and the bovine first version is mainly aimed at the parentage verification. As well, it's for various reasons, but this was the most practical start. Just to give you some background on the scientific part, although time is running out, so I'll probably skip most of this. It's kind of the standard approach based on a genome. You align the sequences that you have, set up the assays and run them in the lab. Sometimes you have to do multiple assay designs because they are tough to work with. And of course, some of the long homozygous stretches are impossible to run on most technologies anyway. So it's going to be a situation where 98% of the markers will be running on this platform and the 2% remaining markers will be done on other platforms, including the capillary electrophoresis. This kind of standard definition, but it's useful for the next slides that I'll show you. One is the sample call rate, which is really important for our clients. And one is the marker call rate, and the combination of the two are really important for a routine lab. This is kind of representative for the next level of equipment, because if you don't get the chip loading right, you will not get good quality results because you have too many incomplete cells. It's typical to get about 60% of the total loading capacity on a useful basis, which is pretty good of what we've seen in our own routine. Because of the design approach, we tend to find around 150 to 200 base pair sequence length. And what is really useful in our field as well, that we automatically detect a number of de novo variants that had not been described before. And that's not especially in our interest, but it opens up the possibilities to work with universities who are who are interested in those extra variants, and then we have another useful cycle of networking and collaboration. And I showed this result, this slide, just to mainly to focus on the left part of this, which means that 99% of the results that we actually find in the diagnostic setting align to where they are supposed to. Again, just focusing on the quality of what we need for our routine applications. And of course, this is a little. We're only showing you the good results, but in the majority of things, this works perfectly, at least better than what we are used to when we do individual reactions or other platforms. And the reason for that is, again, we do focus on the high coverage, and that really gives you very good results, which would not be unexpected. This actually is one of the examples that we did last year on the pilots, and you would probably, I don't know if you can see that actually, but it's ridiculously high coverage. But just to show you the overall call rates across the markers, and this is without any further tweaking. This is just the first assay design. So what we currently have is we're running in parallel with our own routine at the moment, with our historical routines, and I don't have those data yet at the moment. I just wanted to show you some examples that we are pretty confident that this will work across most of our requirements as we see them to grow. So we currently have a first feline panel of about 60 to 70 genetic variants that go across the board. We have between 150 and 200 running markers in the canine panel at the moment. And the current bovine panel is based on the international standard for SNPs and parentage verification and also available. And it's fine that that one is a little bit lower on the call rate because some of those markers have been coming from less well-defined backgrounds. So we expect that call rate to be a bit lower. But on average, across the board, robust performance. So we kind of look at this as a short-term development as well as a long-term co-development. What we want to do with this is have this up and running on the service and through our distributors across Europe at the moment, where at the same time some of these products will be available through the larger Thermo Fisher Scientific, which is again the innovative cycle that benefits us as well as the global market, because we're too small to go global, but you would expect that from us. And what we want to do on the short term to make sure that these panels are as complete as they can be at the moment is to do one more round of panel expansion before it comes out. But that's the short term. On the longer term, we want to take this into the next, and I thought it was an extra slide in here, that's why I go to the long-term stuff. We're currently working on the next level of these kind of V2, I would say, where when the primary panels have been filled up, we want to add specific panels for both parentage as well as genetic variants. And kind of the idea is on the long run to have one tube approach for every animal that for a particular species comes into the lab for diagnostic requests. And there's multiple other applications here where we focus on, but they're on the list for much longer term development rather than short term development. So my conclusions, there you go. What we have seen until now, it's a very robust system in the way we work with it at the moment, and that's very good for us for the future, and I really hope that that's going to be available to the rest of the community as well. And what we have seen until now, it really confirms that you have a large flexibility in the marker panels. There's one or two things to note that, of course, you will never be able to have one platform that allows for all the difficult markers or complex markers to run. What we also have an increasing debate is actually with some of the publications, the data that they are giving out is incomplete or inaccurate, and you only identify that when you start running these kinds of marker panels. One of the things we are currently focusing is that it seems that there's a few papers out where people have been using them a lot on the diagnostic side, but it might be that it's an incorrect research, and that really brings some of the ethical discussions to this field as well. So it's kind of an interesting but complex field. Also, where this is really useful is in our plants that's kind of aimed at resistance genes or fixed panels across different species, like tomato panels, pepper panels. And then of course it would be possible to use a fixed panel and then add on with some of the proprietary markers that you would like for your own project. So we like to take this to the next level. And what's really useful, especially when you have fixed marker panels, that you have a lot of flexibility in the sample numbers, because we can use the 384 different barcodes across the different species and the different applications in the same analysis run. So for us, it means that in real life, this is going to help us to realize fast turnaround times, which is important for our local European network. It's, again, the labor issue. It's low on the hands-on time, and because of the high level of automation, it's really consistent and working well. And for us, what we've seen in the routine lab, but that's also because of the partnership, maybe, it's really plug and play if you have a high-quality lab. That's kind of what I would summarize it to. So with that, I would like to close this presentation, and thank you for your attention.